Hello and welcome to Research Matters podcasts. Today Aishwarya Viswamitra, will give you a summary of science stories that were published during the week on Research Matters. In your weekly dose of Indian science news today, I cover stories on neuroscience, ecology, health, and geophysics. Let's start off this week with a bang. To be precise, the explosion caused by a nuclear bomb. Today, there is widespread race among many countries to acquire, develop, and test nuclear arms, in spite of the devastating effects such bombs have had on the cities of Hiroshima and Nagasaki. Many countries like USA, China, Russia, and India possess nuclear weapons and periodically develop new ones and test them. North Korea is one such country which is secretive about its arsenal, and recently, in 2017, it tested its most powerful nuclear weapon in Punggye-ri. Although it was known that this test was successful, there was little hard evidence on the size of the bomb, the resulting explosion, and the energy it produced. In such cases, scientists use ground-based equipment that measure the tremors produced by such explosions in the earth and derive these parameters. However, since that was not possible in North Korea, there were conflicting measurements that arise due to satellite-based data. In a recent study, scientists from the Indian Space Research Organization, ISRO, use satellite-based data from interferometric synthetic aperture radar, or INSAR, to understand more about the explosion with higher accuracy. They use data from two satellites and a mathematical technique called Bayesian modeling. They found that the explosive power of the nuclear bomb tested was about 245 to 271 kilotons. To put that into perspective, 17 times stronger than the bomb that decimated the city of Hiroshima. The study honed down this number from a broad range of 100 to 700 kilotons, reducing the uncertainty by as much as 97%, and estimated the resulting depth from the explosion with better accuracy. While we have been able to resolve uncertainties in distant nuclear explosions, humanity is yet to understand some of the things that happen in our brains. One of them is the changes that occur in the brain when we read some letters of a familiar language. A new research by the neuroscience researchers at the Indian Institute of Science, Bengaluru, may have some insights into what changes happen in our brains when we read that can help us to be fluent at it. The researchers, for the first time, used two Indian languages in their experiment, Telugu and Malayalam, as they have some similar sounds but different scripts. Two groups of people, one who could read Malayalam but not Telugu, and the other who could read Telugu but not Malayalam, were given some tasks. They were asked to search for an odd letter string in a field of identical letter strings, a game of pick the odd one out. Although this task could have been done without knowing the language, the researchers found that the participants who knew the language were not only better at identifying single letters of it, but were also better at identifying multiple letters. This is because, for those who knew the language, the letters stood out differently, rather than as some squiggles, suggesting that this change in visual processing by the brain enables fluent reading. The researchers also measured brain activity during the process of reading, using function magnetic resonance imaging, or fMRI, which could identify the brain regions responsible for visually processing words and reading them. They found that brain responses to words were better predicted by letters in the lateral occipital region, which is essential for object recognition. While we are in the process of knowing our brain, let's leap to learn a bit about our pancreas and the associated condition, diabetes. Diabetes results when the pancreas either does not produce enough of the hormone insulin or when our body does not effectively use it. As a result, there is an elevated level of glucose in the blood. About 463 million people over the age of 18 worldwide live with diabetes. If the level of blood glucose is not regularly monitored to keep in check, it could result in potentially life-threatening complications. The current method to measure blood glucose concentration is to use a sensor on which a drop of blood, bled by a needle prick, is placed. The sensor contains an enzyme called glucose oxidase, which reacts with the amount of glucose and indirectly measures its concentration. This method, although can be done at home, involves a needle prick each time. How about a pain-free alternative? In a recent study, researchers at the Indian Institute of Technology, Indore and Bombay have developed a biosensor that is as thin as a human hair. It contains alginate spheres with platinum porphyrin and glucose oxidase. The platinum porphyrin absorbs light radiation in the visible region and re-emits in the near-infrared region. In the presence of oxygen, the intensity of the re-emitted light by platinum porphyrin changes as the glucose oxidase reacts with the blood glucose. Since these alginate spheres are covered with the biopolymer, they are also non-toxic to our body tissues and hence can be used to make implantable biosensors, like a tattoo. They can be embedded in the skin and blood glucose can be measured regularly without the need to draw blood. While this sounds exciting, it might still take time before it is available in the drugstores close to you. Did you say you could not wait for such sensors? Well, unfortunately, clearing something for medical use by humans is not easy. Take the example of drugs. Getting a drug from its discovery to commercialization is a laborious process. It involves identifying the right molecules, the mechanisms involved in the body, possible side effects, rigorous testing on humans and non-humans, regulatory approvals, and finally commercialization. The first step, identifying targets, is quite a trick. In a new study, researchers at the Indian Institute of Science, Education and Research, Pune, have used computational approaches to identify such targets to help develop a cure for the deadly Nipah virus infection. The Nipah virus is an RNA virus, meaning that its genetic material is made up of RNA instead of DNA, like that in most animals. The virus RNA is encapsulated in a protein envelope made up of 6 types of proteins. The virus produces 3 more proteins to defend itself from the immune system of the host it attacks. Any disruptions in its ability to produce these proteins or their functions, then the virus cannot proliferate. Thus, any of these 9 protein syntheses can be targeted by a drug to stop the virus. In this study, the researchers used the genetic sequence of the Malaysian strain of Nipah virus to construct a computational model of their protein structures. These models were then used to design peptide inhibitors, which are short chains of amino acids that could interfere with the molecular mechanisms of the viral proteins. Let's break this down. For example, one of the virus's structural proteins, called the G protein, binds to receptors in the host cell in order to gain entry into the cell. The 2 inhibitors the researchers designed for the G protein were intended to disrupt the binding process. Thus, the virus will be unable to hook onto the host cell and hijack its molecular mechanism to replicate. These protein targets and the computational model the researchers hope can be used by drug companies to make drug molecules that fight against the Nipah virus. Yes, we spoke about a long road to drugs, and now how about the shortest path? Did you know ants optimize their path between a point of origin and their destination? Many of them use chemicals called pheromones to lay out a trail and choose the shortest path. However, what happens in ants that do not produce pheromones? Do they still choose the shortest path? Yes, says a new study by researchers at the Indian Institute of Science, Education and Research, Kolkata. The researchers have observed the behavior of the Indian queenless ant, a species native to the Indian subcontinent where they relocate their nest. During the process of nest relocation, these ants use tandem running, a process where some ants lead the rest of their colony from the old nest to the new nest. The leading ants are called tandem leaders, and the trailing ant continually touches its leader's legs and abdomen with its antennae until the pair reach the new nest. The tandem leader then returns to the old nest and guides the next follower ant. As fascinating as it sounds, this relocation is not without challenges. Temperature, humidity, predators, and thieves who steal the brood all pose a threat to the survival of these ants, who are at the most vulnerable when they are shifting the nests. In their study, the researchers wanted to test if the tandem leaders could choose the shortest path among the available ones. The researchers designed several experiments that included paths of various lengths between the old and the new nest. They found that when 2 routes of different lengths were given, the ants chose the shortest routes 60% of the time. When given a mix of short and long routes, the tandem leaders explored all possible options and most of them picked the shortest path to relocate the colony. So far, we do not know how these ants, which do not lay a trail of pheromones, find the shortest path. But the researchers believe that sensory systems like visual, tactile, and olfactory are involved. In the next set of experiments, they plan on impairing these sensory systems to test the theory. Thank you for listening to this episode of Research Matters podcast. See you next weekend. For more such podcasts and news, please log on to www.researchmatters.in.